Okay, so good afternoon. It's a pleasure to introduce Professor Vishwesha Guttal from the Indian Institute of Science in Bangalore. So Vishu, as he's popularly known as, did his PhD from Ohio State University and postdoc at Princeton University. And after that, I believe he moved back to IISc. And he's currently a professor at IISc doing very interesting work on tipping point dynamics and eco-evolutionary dynamics. So it's wonderful. He's also one of the co-organizers of this meeting. So it's wonderful to have him represent the organizers as a speaker.
Thank you very much, Supratim. Although he said I'm one of the co-organizers, that the amount of work I have done is not more than a few emails. All the credit to Supratim and Sagar, who have done a really excellent job putting together a great line of speakers and doing all the necessary background work that got you together. So thanks guys, and I hope you're also enjoying the really brilliant set of talks here. So today I will talk about eco-evolutionary dynamics of finite populations. So in some sense, so there are elements of what I'm going to present today that sort of naturally continues some of the ideas you sort of heard this morning, and using a similar framework, but some very interesting results in addition. Okay, so broadly, before I go to the specifics of my talk, let me emphasize that I do not really study evolutionary game theory for living. So our work in our lab on evolutionary dynamics is sort of opportunistic, as and when various questions we ask about spatial patterns in ecology leads us to ask also some interesting questions in evolution. We study broadly collective behavior and self-organization in a variety of organisms. With collaborators, we have studied cell migration. In our own lab, we do experiments with fish. We do fieldwork as well in grassland, studying blackbuck herds. And we also look at really large-scale ecosystems and ask what are the patterns that we can explain, coarse-graining various details at the level of individuals. And we try to understand mechanisms of these patterns, functions of these patterns, using theory, simulations, experiments, as well as observational data. So this is broadly how I have planned today's talk. So I'm going to present some very recent work on eco-evolutionary dynamics in small populations, where I will describe a general formalism for treating finite population dynamics. I will not go into mathematical details, but what I will try to convince you is that stochasticity in small fluctuating populations can actually have results that would be exactly opposite of what you would have anticipated from natural selection. In addition, this is going to be the main focus of the talk today because that's relevant to the work of this workshop. In addition, I'll also give a sort of brief advertisement that sort of covers evolution of collective behavior that we have studied in our lab over the last 10 years. So as I said, we ask these questions on evolution as and when there is an interesting context in the context of collective behavior and self-organization. So that would be really very short, maybe one slide with pictures, whereas the part one is what I will try to go into some detail. So let's start with the simplest case of evolutionary dynamics. Let's assume there are two traits that are competing with one another. So I'm representing these two traits by these two colored birds. And as you know, there are births and deaths that happen that changes the frequency of these traits, right? So in this specific example, the number of blue birds increased. The proportion of blue birds have increased. So now how do we keep track of these proportions? You learned already several times over the last 2 days. If we want to keep track of, let's say, P is the proportion of type 1, which could be blue birds, how does this change? you would all know by now, that follows replicator equation, right? So dp by dt measures how this proportion of types change, and if there are only two types, this is how the replicator equation looks like, where omega 1 and omega 2 are basically the fitness of two types. And omega itself is basically the per capita birth rate minus the per capita death rate of each of those types. So this is something I think we have now seen probably 4 times at least, if not more, in the last 2 days, right? So let us see what happens, in the limit of infinite populations, as Christian very nicely explained this morning. Replicator equation assumes infinite populations, right? So that's when you see the effects of natural selection. So the classic sort of statement, survival of the fittest, you can very easily see that with the replicator equation. For example, if the fitness of the blue type is more than that of the red type, omega is greater than, so this differential fitness is positive, therefore P would basically, P that corresponds to proportion of blue eventually reaches 1, whereas the red types will basically go down to 0. This is something we all know, a very straightforward consequence of natural selection. But we also learned this morning about the fact that in finite populations, there are stochastic effects. Let's consider the extreme case here where we have two types having equal fitness. So the fitness of blue is same as that of red. So that's when you have the classic genetic drift, right? Because there are stochastic fluctuations, even though there is no selection, the proportion of the two types will keep changing. So I am illustrating two simulations here, showing that in this case, although I started off with exactly half-half of the two types, in this case, the red actually undergoes a fixation, whereas in this case, the blue undergoes fixation, despite the fact that the parameters were all same. Okay, and the classic result in genetic drift is the probability of fixation of the red type is equal to initial frequency of the red type. Okay, so these are the classic textbook stuff that we all study when we study evolution in a mathematical way, right? So now, however, much of the finite population theory assumes that the population size is fixed. You might recall the tutorial this morning when the Moran process was described. An individual reproduces, right, proportional to its, at a rate proportional to its fitness. However, there is also a death of a randomly chosen individual that follows, and this ensures that the population size is always fixed. And this is often true in most models of evolution that accounts for finite populations. For mathematical convenience, we often assume that the population size is fixed. However, if you really think of the realistic scenarios, because of stochastic birth and death, not only do frequencies fluctuate over time, but even the population sizes should fluctuate over time. Okay, now this simple fact is sort of by and large, I wouldn't say always, but by and large, ignored in classic models. Okay, so how does that matter? Does it really matter whether I sort of keep the total population size fixed, or do I let that also change over time? It turns out that it does. I will show you an example here. So now the moment you allow both the population size to increase and decrease, that's the typical population dynamics. We call those ecological dynamics. When we study trait frequencies, we call that evolutionary dynamics. When we allow both the population size and the trait frequencies to change, we call that eco-evolutionary dynamics. So here is an example of two simulations. I will not tell you the underlying conditions, but let me just tell you that the only thing that is different between these two simulations is that this has a larger population size, larger system size, whereas this admits a smaller system size. Now observe the two graphs here. So when you have a larger population size, basically selection is dominant. And in this case, you are seeing that the red type actually is undergoing a fixation. If we wait for long enough, it would reach there. On the other hand, all else being equal, the only difference between these two is that I'm looking at a smaller population size. You see that exactly opposite. Red is actually going in extinction, whereas the blue survives. Okay. Now, how is this possible? So basically, the point I would like to make in this presentation today is that when you have small populations, if you account for the fact that the total population size actually fluctuates, you can get some dramatically new results that you wouldn't see otherwise. Okay. So how does this happen? So to understand this, so you don't have to read this paper because I'm going to present summary of this paper. So this work was basically done by Chakraborty, who is now a PhD student in one of the Max Planck universities with Hannah Coco, and just very recently published, just 2 months ago. So what do we do in this work? So we start with, to sort of, now by the way, just to make the broader context clear, there have been some simulation studies that sort of find this result in various different contexts. So what we found was that there was a lack of a coherent mathematical framework that explains these results from a first principles that accounts for simple factors like birth, death, stochastically, and then sort of builds up a replicator equation in a sort of a first principle way. That's what we do here. So we start with the assumption that the birth rate of type I can be explained, can be sort of decomposed into population density times the per capita birth rate of type I individuals. A fairly straightforward assumption that is most commonly made. We assume the same thing for the death rates. So death rate, the total death rate of the type X depend on the population density multiplied by the per capita death rate. And then we define two quantities which are again self-explanatory. One is the fitness of type I, and one more quantity which is often not discussed, which is what we call turnover of type I. So fitness is basically birth rate minus the death rate defined at the per capita level, whereas turnover rate basically is the birth rate plus death rate. Okay. Now keep this in mind. I have made a highlighted box here for one good reason, because this has an influence on the evolutionary dynamics. Okay, now here is the mathematical approach. So for the details, you can please go back to the paper. We have tried to explain all the mathematical steps to the extent possible, easy to follow. What we basically do is we start with those assumptions I just mentioned, okay, and then we write down what are called master equation or Kolmogorov forward equation. Probably names you did actually hear in a couple of instances in the previous talks. What this basically does is, for a stochastic process, you have a population densities are changing stochastically over time, right? So what the master equation does is writes an equation for the probability densities, and it tries to write an equation for how that is changing over time. And then we do something called a Van Kampen expansion, which sort of accounts for the fact that the population densities or population numbers are actually finite. Okay. And then we sort of invert the equations to get how population densities change over time. We write a stochastic differential equation, which I will show you in the next step. And then from the population numbers, we actually move to trait frequencies. So trait frequency is what the replicator equation does, except that in our case, we also account for the stochasticity in the trait frequencies. Okay, now all of this mathematical details, please go back to the paper and see. And if we do this, we will get some equations. Let me first explain how does our population dynamics looks like. Sure. So just to be clear, you're right from using the physics terminology of drift. The fitness is the selection term which appears in the deterministic part of the SDE, and the turnover is what appears in the stochastic part, diffusion part of the SDE. The only reason why I say a bit of small caution here, in evolution, drift actually means diffusion for physicists. So apart from the clarification, is absolutely right. Okay. So in fact, this is the equation. So this is how the population density basically changes. So the population density of the type I basically changes proportional to its own density times its own fitness, okay, plus a stochastic term here. This is basically a Gaussian noise, And what this shows is that the stochastic term, okay, actually is controlled by the turnover effect tau, which is birth rate plus the death rate, whereas the deterministic part is controlled by the fitness, which is birth rate minus the death rate. Okay. So now what this basically means is that the expected change of population density is given by the fitness, but the variation in the change in the densities is given by the turnovers. Okay. Now, but in evolution, this is what ecologists usually do. You write down equations for all the population densities. But evolution, population geneticists want to write down equations for the trait frequencies. So we basically do a variable transformation from X to P's. P basically represents the proportion of types. If you do that, this term is, so this is the Gaussian noise term, which has a variance dt, which is standard, Wiener process. Okay. Again, if you want the mathematical details, I am intentionally skipping. Usually, you would expect a variance 1. Is that what you're thinking? For a but continuous time process, the variance is usually dt. Okay. Oh, why? So it is just that every one of them has their own Gaussian white noise, but the mean and the variance is same. That is right. Okay. The only difference is the fact that the DWI for the I and J are different. They are uncorrelated. Okay. So now we do. Multiplication by dt in the variance. Of dxi is tau I xi by k times dt. You're right. You're right. Yes. Yes. Thank you. Yeah. Okay. But by then you would also have a dt here. Okay. Both. So basically, I have. Okay. Yeah. Thanks. Okay, so now let us look at the stochastic generalization of replicator equation. The moment you go from population densities to, the timer was not set, so I don't know what's the time. So I have 1 hour 25 minutes. So, okay, I get some bonus points, bonus time. Okay. So if you go from population densities to trait frequencies, you get the replicator equation, right? So let us now make a brief recollection. This is the standard replicator equation assuming no stochasticity, right? dpi by dt is omega I minus omega bar times pi. Okay, that means that if omega I is greater than the average fitness, then that type will increase in population. This is all we know. I'm recapping it just for make sure we all remember. This is the stochastic version that we get after you convert the densities to trait frequencies using what is called Ito's lemma. This is how it looks like. So let me just take you through each of these terms one by one. Look at these two terms first. The last term is the noise term. So again, this is a Wiener process, a standard Gaussian white noise. Okay, and in the limit of population size going to infinity, this basically doesn't exist. Again, there is one more term here. This term is in the physics drift term, or the selection. It's part of the deterministic term. Okay, and this term also doesn't exist in the k is equal to infinity or large population size limit. So basically, if I say take large population size limit, I get the same equation. So that means I haven't done horribly wrong. Okay, but these two terms are actually present and important when we have a small population size. This is what we term as the indirect mechanism by which stochasticity affects the evolutionary dynamics. Now, for the purpose of this talk, I will not really go into details of how this term affects. This term can also really change the direction of evolution, but I will not explain for this talk, because for some of you who know how the multiplicative noise works in SDEs, even though the average is 0, multiplicative noise can have very non-trivial effects over long time scales. But for the purpose of this talk, I am going to ignore that entirely. I am going to focus entirely on what is called direct mechanism. Okay, so here is the interesting part, right? So let me focus a bit more on this. Okay, if we only look at the deterministic part here, this is the effect of natural selection. If omega I is greater than the mean, that frequency must increase. But look at this, there is a negative sign here, which is actually pulling the frequencies in the opposite direction. And this is present only for small population sizes 1. And when does this actually act? Whenever the tau I, which is the turnover rate, the summation of the per capita birth plus per capita death rate, if that is less than the average, okay, that rate will actually increase. Okay, so whereas this term tries to increase the fitness, this term tries to reduce the turnover rates. Okay, so understand this, let us consider the simplest possible scenario when there is no selection. Let's assume there are just two alleles like the red and blue. Okay, let us assume finite and but with no selection. So in that case, classic expectation, if you were to use Moran process, would have been that the change in frequencies is basically a Gaussian white noise, right? On the other hand, that is true only if the population size was fixed. But the moment you allow population size to also vary, which is the most natural thing to actually be doing, right? You actually find that the changes in the frequencies is now -1 over n, which is the finite population size effect times the differential of the turnover rates between two types. What this means is that if a turnover type is lower, that actually those frequencies will actually increase. And this is an effect that is something you don't often see when we discuss what is the role of stochasticity in small population sizes. And that is precisely, okay, just to again recall what exactly is turnover, you can think of turnover as actually a pace of life. Consider two types. They may have same fitness, that means they have same number of offspring and they may have similar death rates, therefore they have similar fitnesses. Okay, but however, imagine that their births and deaths are such that one of them is rapidly reproducing and also rapidly dying, whereas the other one is very slowly reproducing and very slowly dying. If these two were to compete, even if they had the same fitness, the one that actually grows slowly and dies slowly, that actually will win the evolutionary competition between these two types. That's what basically this previous equation captures. Okay, and that's precisely why in these graphs, just by changing population size, I was actually able to invert the outcomes. So basically the blue has a lower turnover rate compared to red in these simulations. So although red actually has a higher fitness, okay, because I changed the population size, the effect of turnover rate dominates that of the fitness, and therefore the evolutionary outcomes can be exactly opposite of what you would think it should be. Okay, and so basically the key predictions of our framework is as follows. Outcomes of larger and smaller population need not be same if turnover rates are not same, and this is something that is often ignored in most evolutionary frameworks that tries to address stochasticity. And in smaller populations, slower paced life has an advantage even if fitness are equal, even if fitness are actually not equal. Okay, and here is one more interesting consequence. We often define neutral evolution, right? A textbook definition. Neutral evolution is when two types have equal fitness, right? Because then their probability of fixation is equal to their initial frequency, right? What our sort of formalism shows is that that is true only if the turnover rates are also same. Okay, so when you define neutral evolution, we not only have to assume that their fitnesses are same, we also have to assume that the turnover rates are same. Otherwise, their fixation probabilities actually are different, and in fact, one may actually win over the other. Okay, so that's basically the point of this paper, where we basically show that in finite populations, if you incorporate ecological dynamics to evolutionary dynamics, what I mean by that is, if you account for the fact that population sizes can fluctuate in addition to trait frequencies, then you can actually observe a reversal of evolutionary outcome as compared to selection. Okay. So for more details, for more mathematics, you please look at this paper. And what we also do in this paper are a couple of additional calculations for those of you who are familiar with Price equation and also Fisher's fundamental theorem of evolution. We also derive how those are modified in the presence of ecological dynamics and for finite system sizes. Okay, so that ends the first part of my talk. I still have 1 hour 25 minutes, so I can go on to my second part of the talk. How much time do we really have though? Okay, okay. So I will go over this really quickly. I don't intend to go into a lot of details here. So let's hope this video works. So as I mentioned to you in the beginning, in our lab, we are very interested in understanding how animals form self-organized patterns and why do they form, right? And there are many evolutionary reasons, adaptive reasons why animal groups form these kind of fascinating patterns. Okay, so over years we have studied how migration, cannibalism, predation, cooperation, and even mating, all of these different ecological factors can promote collective behavior and self-organization in a variety of taxa. So, in fact, before we started, how did I actually, if I were to think, why did I get into this previous study to look at finite size population effects? The precursor to that actually was this study by Jaideep Joshi, who is now a faculty member in IIT Bombay. He started looking at the dilemma of cooperation in animal groups. Typically, when people study cooperation, we study using the simple payoff matrices, assuming well-mixed populations and large populations, right? So here we started to look at, I want to look at cooperation in organisms that actually have mobility, okay, and which actually form a variety of group sizes. Typically, you think of cooperation in two-player games or n-player games, right? But if you look at animal groups, they occur in all variety of group sizes. Some in the same species, some groups might be of 5 individuals, some groups might be of 100 individuals, right? So in these kind of very varied contexts, how can cooperation emerge? And we were really inspired by some of the work done by Sylvia and her student Garcia. And we realized that one of the key assumptions behind mobility, why do people not look at mobility, is that mobility destroys the assortment that is required for cooperation, right? So I think in Shitabra's talk yesterday and as well as one of today's talks, you heard that for cooperation to survive, cooperators must somehow segregate themselves and move away from defectors in some way, right? Whatever the mechanism that might be. It might be punishment, for example, right? By punishing, I sort of, eliminate cooperators. It could be like I literally move away from cooperators. So you need some kind of assortment of cooperators for the cooperators to survive. Okay, but typically you think that mobility, like these fish groups, which show a lot of mobility, those usually kill assortment because they're constantly mixing with one another, right? So can cooperation emerge in those kind of systems? So that's the question that Jaideep led to sort of address. And we find that in fact that need not be the case. Even with mobility, it is possible that cooperation can evolve. And the mechanism was actually inspired by some of the work by Sylvia, which showed that if there are differential adhesion between individuals, cooperators can spontaneously form a different set of groups than defectors. And this can, what we showed in this, no, this didn't work. How do I make this work? There's an animation here. You have to imagine things moving. Maybe I have to click here. Okay. So here I'm showing you a simulation of animal groups moving, where red types you can think of as defectors, blue types as cooperators. If they have same amount of adhesion, every group will have both cooperators and defectors. In the second part, we assumed that the numbers were different, right? That indicated that they have different degree of adhesion among themselves. You can now see that naturally the cooperators, which are indicated by blue here, will segregate into a different group, even though individuals cannot recognize other individuals. Okay, so this sort of is a work that where we used evolutionary game theory, but applied in the context of a large number of individuals who are moving in space and interacting with one another. And these are not not well mixed populations, right? Their structure is changing over time in a highly dynamic way. And then in another work, what we showed was that predation can actually favor collective dynamics. Now, if you think about the classic theories of why do animal forms group, predation is often the main reason that is attributed. Yeah? Yes, in those models, yes. And in fact, they actually show cycles, cyclical behavior. So defectors keep trying to copy the behavior of cooperators, and cooperators try to sort of continuously keep updated with different strategy. Yeah, and they show cyclical behaviors. Cooperators then collapse, and they again emerge And what we show in one of the papers is that re-emergence really requires that the groups formed are small. So therefore, accidentally, the small groups that form will all have cooperators with a much higher chance than a large group that forms will have all cooperators. That stochastic effects are actually what amplifies the emergence of cooperation. Predation is a classic hypothesis to explain why animals form groups. Now imagine we are all animals. There's a man-eater who just suddenly pops in here. Okay, who has the highest chance of getting eaten? Me, because I'm away from all of you, right? So basically, because the region around me, which is sort of empty and is open for attack, is highest for me. So for me, the best strategy, therefore, should be to come to the center of this entire room, right? So that's the idea called selfish herd proposed by Hamilton. But that doesn't quite explain why animals move in a synchronized fashion. It only explains why they group. So what we did in these experiments was displayed simulations. These are all simulated particles. They have different types of collective behavior. This is a swarming particle. These are all highly polarized particles. This is a solitary particle. What you see here is a fish that comes from the other side of the tank and thinks these are the prey, and they keep attacking. So what we basically did was we counted, whether the solitary guys were attacked more or were the ones who were moving in these kind of groups which were attacked more. So we just kept track of how many times each of those dots were attacked. And what we found was that the solitary ones had the highest attack rates, followed by the ones, which are highly polarized. So it's the intermediate ones who sort of wiggle around a lot. They had the least risk of attack. That suggests that when organisms are together, there is a dilution of the risk. In addition, when they move in a slightly stochastic way, there is also a confusion that the predators face, and therefore they have a hard time tracing and attacking them. In another work, what we showed was that selection for migration can spontaneously lead to both leaders and emergence of cooperation. So in these simulations, what we did was, in the beginning, we assumed that everybody is solitary and there are no leaders who know where to go to migrate. But we assume that if somehow you figured out how to migrate, you will have a higher fitness. Now, what happens as a consequence is that, it turns out that I don't have to be a leader myself. I just have to copy somebody around me. If there is a very good chance that somebody around me might actually know where to go for migration, and that sort of that selection leads to emergence of some small proportion of the entire group being leaders, which are represented by blue dots here. The green dots are all individuals who are blindly following their neighbors, but yet the entire group manages to migrate large distances. So this is a very recent work that we are currently doing, where we are studying the mating patterns in black bugs. So what you are seeing here is another type of collective behavior. Basically, these are all territories marked by highly territorial males. So unlike all the fish and other organisms I was showing in the past, where they were coordinating their behavior, here they actually don't want to coordinate their behavior. They're highly territorial. But however, once in a year, they all aggregate in one place. This is a Velavadar National Park in Gujarat, and there is one specific spot in that national park where they all aggregate. Roughly, actually, this season, as of now, I got a call saying that currently they are doing this display. They all aggregate in this region, and they fight to get the best spot in the entire area, because females are more likely to come and mate if we have the central location in the entire area. And these are called leks. So leks are basically a very interesting phenomena where everybody is trying to maintain their space, but they still aggregate together. Okay, by doing so, they actually increase their chances of mating success. So what we are trying to do is to study the interaction between males and males, females and females, and males and females on these leks. You can see lots of fights are happening there, and lots of chasing is happening between females and males, as well as males and females. And only some of them lead to eventually mating behaviors. We're trying to now do modeling as well as characterization of these interactions using these drone-based footages. Okay, so with that, so this is a short summary of various work we have done to understand the evolution of collective dynamics, and summary of the work on finite size evolutionary dynamics. Okay, the key result here is that in small populations, results can be very counterintuitive, and that is driven by turnover rates. Okay, so with that, I would like to thank all the funding agencies and various students who have done this work. Thank you.
Can you display the stochastic differential equation again?
Yeah, sure. Yes.
The last term doesn't have anything proportional to pi?
Yes, have not mentioned. So this does have, the variance of this does depend on p. Yes,
So otherwise it will go negative.
Yeah. So it actually is proportional to p into 1 minus p sort of terms. Yeah. Thanks.
Yeah, thank you for the talk, sir. I just had a question driven out of curiosity. The slide where you showed the fish is going towards the isolated dot. Like, how was that setup achieved? Like, what are the dots exactly?
So the dots,
Like,
so usually a lot of organism this size, so many, for example, let's say if you have a cat at your home, or if you have a lizard, if you have a laser pointer, if you display near them, many of them have a tendency to go and catch them, and that is true for fish also. And in this case, the dots we think are some representation of something that looks like their natural prey. Okay, of course, how do we know for sure? We don't know. But if you look at the action that they do when they go near the dot, like a sucking action, which is very typical of trying to eat something, seems like, the dots that we have projected, that mimics their predatory behaviors.
got it. Thank you, sir.
This reverse process that you showed, so of course these trajectories that you showed probably are just one single trajectory, right?
Yes.
So the question is that how rare is this event, or how often can one expect such things to happen?
So for this, in the paper we show very clearly that we can also analytically calculate the steady state PDFs. Okay, and then we find that it is consistent with these single realizations. So basically, when the population size is large, the PDF would be, would show that the red type has a very high frequency when the population size is large, and when it's the opposite, it would be exact. The PDF peak would be skewed towards red frequency is close to 0.
Oh, so full distribution.
Yeah, absolutely. We can even calculate we can obtain the semi-analytic approximation for the PDF.
Yeah. Okay, thank you.
Thank you. Sorry, if I understood correctly, in order to obtain your equation for the frequencies, you start from the equation on the abundances, and then
yes.
you derive an equation, I guess, for the total abundance.
So this is the equation for abundance of each of the type.
Yeah.
And then this describes the entire system completely, right?
Yeah.
So then I basically look at xi divided by summation of xi's.
Yeah.
And then I use Vito's lemma to compute equation for dp. But you are right that there has to be now one more equation, right? Because there were n variables.
Yeah.
Now there are n-1. So we also have an equation for the total population size, which I did not write here. Sorry. Yeah. Thanks for pointing that out.
Yeah, because it appears as a
there is an equation for
non-autonomous term.
nkt.
Yeah.
There's a differential SD for NKT as well, yes. Which basically, if I remember correctly, DNK is proportional to NK times the average fitness of the entire population. Plus
Thank you.
noise term.
So there was a question
Yeah.
on this side.
But he has the authority. Okay.
Hi. Sorry,
You go ahead, ma'am. It's okay. You had the mic.
you had mentioned that neutral evolution would probably then require these traits to also have similar turnover rates.
Equal turnover rates. Yes.
Yeah, equal turnover rates. That sort
Of course, it's a bit debatable. One can also say, I would call something neutral if this entire term is 0. But in the paper, we took a stand that independently they must be 0.
Okay. I was thinking about gene drives.
Sorry?
Gene drives.
Gene drives, okay.
Yeah, so essentially it was traits which are able to go to the next generation more than half the probability because they're able to sort of stick around. I don't know why. So they're typically used in like controlling mosquito populations by injecting them with things which reduce their mating chances. So which essentially reduces their population size. But it has so far been it's not been very useful. So if we sort of have this information that if we want something to go to fixation, it needs to have a lower turnover rate. Then we essentially could find gene drives that spread quickly but do not increase the death rate enough so that they actually get through the entire population rather than just dying out.
Okay. Just to be clear, the slower turnover rates means higher chance of fixation.
Yeah.
Yeah. But I see a very interesting thought. I think
Yeah.
we can discuss more. I think there are people who study mosquitoes and other things.
Yeah, absolutely.
Might be actually able to comment more usefully than I can.
Okay.
Thanks, a good
Can
point.
you,
Yeah.
Can you hear me now?
Yeah.
Okay. So, if I understand correctly, each SDE is corresponding to a single type. So, what was the typical polymorphism in your system, and how did you sort of draw the new birth and death rates for each new type that you were creating?
This one.
Yeah. So, let's say
In this type, there is a more general equation in the paper which accounts for mutations. But in this specific equation, there are no mutations. So whoever is present, and the number of types that can be, discrete types that can be present is arbitrarily large. There is no limit on that. Right. I just showed you the example of 2 types. It helps you with the intuition.
So, did you do simulations with potentially infinite types? Because that's
no, we didn't.
Okay. Because we've been having problems with that, so I was wondering if you had like a solution.
Okay. So here the sort of focus was the derivation of the equations and then testing a few special cases like 2 species types.
Thank you.
Thank you. Thank you.
Thank you. We break for coffee and we can continue this discussion over coffee. Thank you so much.
Thank you.